This is looking at microbial growth. This corresponds to exercise 37 in your lab manual. To study growth, you need to try to understand what some of the requirements are, and to figure out optimum growth, you need to have optimum pH, the optimum temperature, all of the appropriate and necessary nutrients and supplements, and then also what is the oxygen level. Some organisms will need a high concentration of oxygen. For some organisms, oxygen will actually kill them. And so you need to know what your organism's requirements are to get optimum growth, or if you are studying a brand new organism, you're going to have to figure out what these optimum requirements are. Bacteria, remember, grow and reproduce by binary fission. When we talk about bacterial growth, we're talking about the increase in the number of viable cells. And along with that, we often calculate what is the generation time. The generation time is the time that's needed for that population of microorganisms that you have to double in cell number. So what is the time for it to go from, say, 100 cells to 200 cells, or 2,000 cells to 4,000 cells? You want to know what that time frame is. And that's going to vary depending on what species you're looking at. Some organisms, the generation time may be 2 hours. Others, it may be 30 minutes. E. coli, one of the most well-studied bacteria, we know everything about it. We know what its optimum growth requirements are. So under the best conditions, the generation time for E. coli sometimes can be as short as 18 minutes. So it's going to vary depending on the organism you're talking about. There are 4 different growth phases for bacteria. There's the lag phase, the log or exponential phase, stationary, and finally the death or decline phase. And that is what is shown on this diagram here. The order of which they occur, as you can see, lag phase, you're not really seeing any cell division at this time. Usually it's when you first place the organism into a new medium, it's adjusting to that. The cells are increasing in size but not number. And then the log or exponential phase is where you have a huge increase in the number of cells at a steady rate. And then stationary phase followed by the death phase.